In this video, we will discuss about insulin biosynthesis and the secretion by pancreatic beta cell, and how this uh, glucose helps in generation of energy. So we will start with the insulin. So what happens is the transcription of insulins happens in the nucleus as for other genes, just like other genes. After that, the mRNA is translated into the pre-pro insulin in the cytosol, or we can say in the endoplasmic reticulum, as the it's a its complete translation happened in the endoplasmic reticulum, but it begins in the cytosol. So after pre-pro insulin is formed, it will then acquire its native structure by forming a uh, native disulfide bond within the endoplasmic reticulum. In the endoplasmic reticulum itself, and then it moves into the Golgi for further post-translation modification or further maturation. When it reaches the trans-Golgi network, it will then directed to the secreted granule, where this pro-insulin is cleaved off in two parts. First one is the insulin, which is the main function, and second one is the C-peptide. And after that, it will move this, we can say, secretory granule will move out of the cells through exocytosis process, and we will understand when this triggering of this insulin into the triggering of this insulin in the extracellular matrix, or secretion by this pancreatic cells happen. For this, we need to understand the glucose function. So what happens when glucose enters the cell through different receptors, such as GLUT1, GLUT2, or in common we called it as GLUTs. So, the glycolysis of the glucose occurs that leads to the pyruvate formation, and after this pyruvate formation, it will then go into the TCA cycle in the mitochondria, where ATP generation is occurred. And whenever the ATP generation is occurred, there is increase in the amount of ATP. So whenever the ATP to ADP ratio increases, this triggers the KTP channel that will leads to membrane depolarization. And because of this, what happens is that it will triggers the calcium influx within the cytosol of the this pancreatic beta cells, and this increase in the amount of calcium within the pancreatic beta cell will trigger the exocytosis of the matured insulin uh, through this pancreatic beta cell. And this exocytosis occurs uh, this exocytosis will occur uh, because of the high CA2 concentration within the cytosol. And this is how the glucose intake will leads to insulin secretion outside the cells, and insulin will then be able to control the glucose level. And whenever there is a decrease in the amount of glucose level, the influx of the glucose through GLUT decreases, and this will decrease the ATP to ADP ratio, and this calcium influx will decrease. And what will happen then is that the calcium concentration will decrease, so the secretion of insulin into the extracellular matrix, or we can say uh, in the blood, will get reduced. So this is how relationship between glucose and insulin works. If you need to understand this thing in detail, or if you need to understand more about this relationship between glucose and insulin, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.